Hello, my dear students. Welcome you all to Sri Jain, the Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, today we are going to start with a very important topic in biomolecules, nucleic acids, DNA, the genetic material, and different other components. So nucleic acids, starting with nucleic acids, F. Friedrich Mischer, he discovered nucleic acid in the nucleus of a pus cell and he named the acid as nuclein. Okay. So Friedrich Mischer, he discovered the nucleic acid in the nucleus of a pus cell and named the acid nuclein. Altman, he later termed that nuclein is nucleic acid. Okay, nucleic acids, they are made up of nucleotides polymer, so it is a polymer of nucleotides, right? Nucleotides, right? Polynucleotide. They contain a nitrogen base, a pentose sugar, and a phosphate in their structure. These three components are there: a nitrogen base, a pentose sugar, and a phosphate group. So one nucleotide contains a nitrogen base, a pentose sugar, and a phosphate group. There are different types of nitrogen bases which form the basis of classification. They are broadly of two types: pyrimidine bases and purine bases. Pyrimidine bases, the structure has one pyrimidine ring which has a skeleton composed of 4 carbon atoms and 2 nitrogen. So there is one pyrimidine ring having 4 carbon atoms and 2 nitrogen atoms. The examples are cytosine, thymine, and uracil. The pyrimidines, they are basically made up of carbon and nitrogen, 4 carbon atoms and 2 nitrogen atoms. They make a ring structure that is known as what pyrimidine ring. Examples are cytosine, thymine, and uracil. Uracil is used in RNA. Okay, uracil is used in RNA. Purine bases, the structure has two rings, one pyrimidine ring, that is 2 nitrogen plus 4 carbon, and other one is imidazole ring. Imidazole ring is made up of 2 nitrogen and 3 carbon. So the pyrimidine ring is made up of 4 carbon, 2 nitrogen, and the imidazole ring is made up of 2 nitrogen and 3 carbon. Adenine and guanine. Okay, adenine and guanine. So in case of ribonucleic acid, in case of deoxyribonucleic acid, the purines are common. The purines, they will be there, they will be present. Okay, adenine and guanine, both in case of RNA and DNA, both in case of RNA and DNA, both in DNA and RNA. In case of pyrimidine bases, especially DNA is having cytosine and thymine, and in case of RNA, thymine is replaced by uracil. Okay, is that understood? So pyrimidine, it is having one pyrimidine ring having 4 carbon, 2 nitrogen. Okay, and the purines, they have two rings. The imidazole ring made up of 2 nitrogens, 3 carbon, and the pyrimidine ring, that is 2 nitrogen and 4 carbon atoms. Right? Adenine and guanine, they are common in both DNA and RNA, and the pyrimidines, in case of RNA, uracil is present, while in case of DNA, cytosine and thymine is present. Understood? Nitrogen bases forms the bond with the first carbon atom of the pentose sugar. The pentose sugar, which is a 5 carbon sugar, on the first carbon, nitrogen base is present, okay, which forms a nucleoside. So nucleotide minus phosphate group is equivalent to nucleoside. Okay, so nitrogen atom in the third place forms a bond with the sugar in pyrimidines, while nitrogen of the ninth place forms bond with sugar in purines. Okay, so the nitrogen atom in the third place, N3, forms bond with sugar. Okay, the pentose sugar, it combines with the third nitrogen atom of nitrogen base in case of pyrimidines, and in case of purines, the ninth nitrogen atom combines with the pentose sugar. The phosphate part forms phosphodiester bond, which is a ester linkage, with the fifth carbon atom of the sugar to form a complete nucleotide in case of purines. Okay. The phosphate parts forms ester bond, it is the covalent bond with the fifth carbon atom of the sugar to form a complete nucleotide in case of purines. Okay, so this is the first carbon, third carbon you can see, this is the second carbon. In the second carbon you can see the oxygen is absent. That is why it is known as what? 2-deoxy ribose, because the second carbon is not having oxygen, not having oxygen. Okay, so the nitrogen bases, they forms bond with the first carbon of the pentose sugar, which forms nucleoside. Means nucleoside is nitrogen base plus pentose sugar. There is no phosphate. Okay, the nitrogen atom of purines the ninth nitrogen atom in case of purines combined with the first carbon of the pentose sugar, and in case of pyrimidines the third nitrogen atoms combined with the first carbon of the pentose sugar, while the phosphate group makes the ester linkage, ester bond, okay, with the fifth carbon of the pentose sugar. Okay, types of nucleosides and nucleotides: adenine, guanine, okay, cytosine, uracil, and thymine. You can see adenine and guanine, they have two rings. Okay, one ring is the imidazole ring, okay, which is a 5-membered ring, and the other ring is the 6-membered ring, which is the pyrimidine ring. Okay, you can see it like guanine, adenine. Okay, then in case of cytosine, uracil, and thymine, there is only one ring with 4 carbon atoms, okay, and 2 nitrogen atoms. While the imidazole ring is having 2 nitrogen atoms and 3 carbon atoms, okay. So these are purines and these are pyrimidines. Adenine plus ribose. The ribose sugar is also a pentose sugar having 5 carbon atoms. The basic difference between ribose and deoxyribose is that in ribose, the second position is having OH group, having hydroxyl group. OH group is present. The hydroxyl group is present. So adenine plus ribose known as what? Adenosine. Adenosine plus phosphate known as adenylic acid. Adenylic acid, AMP. Okay. So the adenine plus ribose is known as what? Adenosine, and adenosine plus phosphate is adenylic acid. So this is the nucleoside. This is a nucleoside, and this is a nucleotide. Okay. Then adenine plus deoxyribose, deoxyadenosine. This is nucleoside. Then deoxyadenosine plus phosphate becomes deoxyadenylic acid, nucleotide. So when the ribose combines with adenine becomes adenosine. When deoxyribose combined with adenine, deoxyadenosine. Okay, adenosine plus phosphate, adenylic acid. Deoxyadenosine plus phosphate, deoxyadenylic acid. Same guanosine, guanylic acid. Okay, then guanine plus deoxyribose, deoxyguanosine. Deoxyguanosine plus phosphate, deoxyguanylic acid. Then the same way, cytidine, cytidylic acid, deoxycytidine, deoxycytidylic acid. Okay, so this nomenclature you have to remember. Okay, but the name, if it is ending on INE, then it is nucleoside, and if it is ending at YLIC acid, then it is nucleotide. Okay. In case of uracil, uracil plus ribose, uridine, okay, and uridine plus phosphate, uridylic acid. Thymine plus deoxyribose, deoxythymidine, deoxythymidine plus phosphate, deoxythymidyllic acid. Okay, understood? So DMP, DGP, DCMP, and DATP all are nucleotides, or when the phosphate is attached, mean they are nucleotides. Deoxyribonucleic acid. Misher, he discovered DNA while the term was given by Zaris. So Misher, he discovered DNA, but the term was given by Zarish. The pentose sugar is a deoxyribose sugar, and the nitrogen base are of 4 types: A, T, G, and C. Wilkins and Franklin, they studied the DNA molecule with the X-ray crystallography technique. Okay, so they study about the structure, the helical model, okay, or we can say the molecular geometry of the DNA molecule with the help of X-ray crystallography technique. Understood? Watson, Crick, and Wilkins were awarded the Nobel Prize in 1962. Watson, Crick, and Wilkins, they were awarded the Nobel Prize in 1962. DNA is made up of two polynucleotide anti-parallel chains, 5 prime, 3 prime, and 3 prime, 5 prime. 5 prime end means the phosphate is free. The phosphate is free. 3 prime end means the OH group is free. Same case, the OH group is free, phosphate is free. Okay. So the DNA is made up of two polynucleotide chains which are anti-parallel. Okay. The polynucleotide chains are complementary as well as anti-parallel to each other. The word anti-parallel means the strands of the DNA are in opposite direction. The word anti-parallel simply means what? That the strands of DNA are opposite in direction of phosphodiester bond. What is opposite in direction? The nitrogen bases, they are complementary. They are complementary. Okay? Means A combines with T with double hydrogen bond and G combined with C with triple hydrogen bond. So if the one strand is having A, T will be there on the second strand. If the one strand is having G, then the other strand will be having C. So that is why it makes the anti-parallel nature of DNA. Okay. So the word anti-parallel means the strands of DNA are in opposite direction of the phosphodiester bond. Okay. If the direction of phosphodiester bond is 3 prime to 5 prime in one strand, then it will be the 5 prime in the 3 prime in the other strand, while the other nitrogen bases remain complementary to each other. Michel, he discovered the DNA while the term was given by Zares. The pentose sugar is a deoxyribose sugar and the nitrogen bases are of four types: A, T, G, and C. Wilkins and Franklin, they studied the DNA molecule with the help of X-ray crystallography technique. Watson and Crick in 1953 proposed the double helical model of DNA with the help of same technique. Watson, Crick, and Wilkins were awarded with the Nobel Prize in 1962. DNA is a polynucleotide made up of two polynucleotide parallel chains, anti-parallel chains as per the mode. The polynucleotide chains are complementary as well as the anti-parallel to each other. In the arrangement of phosphate groups are anti-parallel to each other. The phosphodiester bonds arrangement is also anti-parallel to each other, and the nitrogen bases are complementary to each other. Chargaff's rule. Chargaff's rule says what? The DNA strands are held together by the hydrogen bonds which are present between nitrogen bases of both the strand. Adenine binds to thymine with double hydrogen bond. Guanine binds to cytosine with triple hydrogen bond. Okay. The Chargaff's equivalency rule says in a double-stranded DNA, the amount of purine nucleotides should be equal to the amount of pyrimidine nucleotides. Means the amount of purines and the amount of pyrimidines always be constant for a particular species, okay. So the amount of purines and the amount of pyrimidines, they should be constant, okay, for a particular species. So purine is equal to pyrimidine. A plus G is equal to T plus C. Means A plus G upon T plus C is equal to 1. Purines, adenine and guanine. Pyrimidines, thymine and cytosine. So adenine plus guanine, thymine plus cytosine, they should always be equal to constant, means 1. Base ratio A plus T upon G plus C is constant for a given species. A plus T upon G plus C. Sorry. It is the base ratio is, as we know, A plus G upon T plus C always remain constant. Okay, so the base ratio says the A plus G upon T plus C always remains constant. Okay, it will always be constant. It will always remain constant for a particular species. Okay. Now, the sugar phosphate bond. So here we are having a deoxynucleotide structure, DNA structure, in which you can see the sugar phosphate bond. The pentose sugar, adenine is attached, okay, to the phosphate group, okay, on the 3' end, the phosphate group, okay. The phosphate group combines with the, means this pentose sugar is having OH group on the 3' end, OH group on the 3' end, which combines with phosphate group of 5' and which is present on this carbon. So this carbon, this pentose sugar is having OH group on the 3' end, which combines with the phosphate group of the 5th prime end, or the 5th carbon, not the prime end, 5th carbon of the second nucleotide. So this is adenine and this is thymine. So this is the formation of phosphodiester linkage. You can see it is a ester linkage. Okay, ester means the combination of alcohols and acids. So here we are having acid, phosphoric acid, and the OH group is a alcoholic moiety. Okay, so this is the formation of phosphodiester bond. In this way, the chain is created like A, G, C, T, like that. Okay, is it understood? So the first carbon is having the adenine, the second is thymine, then guanine, then cytosine, and they make the phosphodiester linkage in this way. So this is the formation of deoxyribonucleotide, okay, deoxyribonucleotide which forms. Understood? In a DNA, when A plus T is greater than G plus C, in a DNA, when A plus T is greater than G plus C, the DNA is AT type. If the A plus T is greater than G plus C, then the DNA is AT type. Base ratio of AT DNA type is more than 1. Base ratio of AT DNA type is more than 1. Example, eukaryotic DNA. Okay. In a DNA, if A plus T is greater than G plus C, then the DNA is AT type. The DNA is AT type. The base ratio of AT DNA is more than 1. Okay. So the base ratio which is mentioned here is A plus T upon G plus C always remain constant. Okay. A plus T upon G plus C always remain constant. Okay. Now, in the DNA, when G plus C is greater than A plus T, then the DNA is GC type. Base ratio of GC DNA type is less than 1. Example, prokaryotic DNA. So you can easily, segregate a prokaryotic DNA and a eukaryotic DNA. Eukaryotic DNA, they have A plus T greater than G plus C. Okay, mean the base ratio of AT DNA type is more than 1. Okay, so A plus T upon G plus C is greater than 1. Okay. In case of this, A plus T upon G plus C is less than 1. Okay. Melting point of DNA depends upon GC content, as GC is having triple hydrogen bonding. So it needs high bond dissociation energy. High bond dissociation energy. So if GC content is high, means the bond dissociation energy will be more. Okay, 3 hydrogen bonds which require comparatively more energy for its breakage than AT content. TM. TM is the temperature for melting, temperature of melting. Okay. More the GC content, more will be the temperature required to break the DNA. And it is seen as GC content is more in case of prokaryotic DNA. That is why the melting temperature in case of prokaryotes is more than eukaryotes. Okay, in the prokaryotes, the DNA is provided more amount of heat for its denaturation, for its melting. Okay, for the strands to get unwinded. Understood? The strand of the DNA that participates in the transcription among the two strands is called antisense or non-coding strand or the template strand. Please remember, the strand of the DNA which participates in the transcription. Transcription is the process of formation of messenger RNA from DNA. Messenger RNA from DNA. This process is carried out in the presence of DNA dependent RNA polymerase. Okay, so the transcription is the process of converting DNA into messenger RNA, and out of two strands, the one strand which is responsible for the formation of messenger RNA, which will act as a template for messenger RNA so that the complementary base pairing can happen, that strand is known as what? Anti-strands or non-coding strand. Another strand of the DNA which remains silent during transcription and does not participate in it is known as what? Sense strand or coding strand. Okay. So the anti-sense or the non-coding strand is the strand which will help to make the template, and the another strand of the DNA which remains silent during transcription and does not participate is known as what? Sense strand or coding strand. What is denaturation of DNA? When the normal DNA molecule is placed at high temperature, 80 to 90 degrees Celsius, then both the strands of DNA get separated. They get unwinded, okay, from each other as the hydrogen bonds will break. Between the two strands break, and this is more known as what? DNA denaturation. Means the native structure of function is lost. Is lost. What is renaturation of DNA? When this denatured DNA molecule is cooled or brought down to the normal temperature, then both the DNA strands attached after the hydrogen bonds are formed between both the strands. This is called renaturation of DNA. Okay, this step is taken as a principle for PCR, polymerase chain reaction. Is that understood? Yeah. So when the normal DNA molecule is placed at high temperature, then the DNA molecule is cooled, or the DNA molecule gets unwinded, both the strands they get separated, okay, and the hydrogen bonding will break off. Okay, in the denaturation of DNA, when this denatured DNA is cooled and brought down to the normal temperature, then both the DNA strands is attached to the hydrogen bonds. With the help of hydrogen bonds, both the strands get attached again. This is known as what? Renaturation of DNA. Hyperchromicity. When a double-stranded DNA is exposed to heat that causes its denaturation, and the denatured DNA molecule absorbs more amount of light, this phenomenon is called hyperchromicity. So hyperchromic means absorbing more amount of light. So why it is happening? Because the DNA is denatured. That is why the ends become rough. The surface phenomena will increase, and that is why the hyperchromicity is seen. Understood? Now, hypochromicity, when this double-stranded denatured DNA, when this double-stranded denatured DNA molecule cools down slowly, when it cools down slowly, then return back to the double-stranded form, and it absorbs less amount of light. Hypochromicity is what? When this double-stranded denatured DNA molecule, when cools down, when the renaturation happens, then it will absorb less amount of light, which is known as what? The hypochromicity. So hyperchromicity and hypochromicity is clear. Okay. Renaturation, denaturation, clear. Okay. Now we are moving to the next. Configuration of DNA molecule. The two strands of the DNA are helically coiled. They are coiled helically, similar to a revolving ladder. Okay. Backbone of the DNA is made up of phosphate and sugar, while the steps or the bars are the pairs of nitrogen bases, like this. So the steps are the nitrogen bases, like this. Okay. Understood? The stairs or the steps or the bars are the nitrogen bases, and the backbone is made up of phosphate and sugar. The two successive steps have a distance of 3.4 angstrom. Two successive steps, step number 1, step number 2, they have a distance of 3.4 angstrom. A single complete turn of DNA molecule have 10 steps or 10 pair of nitrogen bases. Hence, one complete turn is 34 angstrom long, which is called helix length. So one helix length is equal to 34 angstrom. Okay, two successive ladders, they are separated by a length of or distance of 3.4 angstrom. Okay, 10 steps are there in one turn. This one turn is having a length of 34 angstrom, which is known as what? Helix length. Diameter of DNA molecule, or the distance between two strands phosphate, is 20 angstrom. The diameter of DNA molecule or the distance between two strands phosphate is 20 angstrom. Distance between two strands is 11.1 angstrom. Okay, the distance between two strands, like these are the two strands, they have a distance of 11.1 angstrom. The diameter of DNA molecule or the distance between two strands phosphate is 20 angstrom. If we compare the distance between two phosphates present on the that strands, two different strands, then it is 20 angstrom, and the distance between two strands is 11.1 angstrom. The hydrogen bonds that are between nitrogen bases are 2.8 to 3 angstrom long. Angle between the nitrogen base and the first carbon is of pentose is 510 degrees. Okay, so the hydrogen bonds that are between the nitrogen bases are 2.8 to 3 angstrom, and the angle between the nitrogen base and the first carbon of pentose is 51 degrees. It is not 510, it is 51 degrees. I will write 51 degrees. Okay. DNA has a molecular weight of 106 to 109 daltons. The molecular weight of DNA is 106 to 109 daltons. The angle between the nitrogen base and the first carbon is 51 degrees. And the hydrogen bonds that are present in between are 3.0 angstrom, 2.8 to 3.0 angstrom. The eukaryotic nucleus have the DNA associated with the histone proteins, okay, which are rich in histidine, basic, okay, which forms nucleoprotein. Nucleoprotein can be easily understood by nucleosome model. Nucleosome model. Okay. The protein histone occupies major groove of the DNA at an angle of 330 degrees. Okay, at an angle of 30 degrees. Okay, so the protein histone occupies the major groove of the DNA. The major groove of the DNA, histone proteins are present that group in that groove at an angle of 30 degrees. The DNA and the histone have a salt linkage or bond having Mg+2 ions. The DNA and histones, they have a salt linkage. Okay, so the histone and the DNA, they are basically linked with the help of salt linkage, okay, or bond in the presence of Mg2 plus. Okay, and they are rich in Mg2 plus, right? Chromosomal DNA is linear while prokaryotic, mitochondrial, and chloroplast DNA is circular. Sinshima isolated the DNA of 5 into 174 bacteriophage and found that it is single stranded. It is single stranded. Okay, almost all the DNAs, they are double stranded. Okay, but there are some few exceptions where single stranded DNA is there. Like Sinshima, he isolated the DNA in 5174 bacteriophage and found that it is single stranded. G4, S13, M13, F1, FD, all are bacteriophages and have single-stranded circular DNA. So G4, S13, M13, F1 and FD, they all are bacteriophages and they have single-stranded DNA. Usually DNA is double-stranded, but in these cases, the retroviruses, bacteriophages, it is, what either retroviruses, they have single stranded RNA. Okay, single stranded RNA. Sorry, retroviruses, they will not come under this category. They have single stranded RNA. Okay. The single stranded DNA are present in bacteriophages like 5 to 174, G4, S13, M13, FD, F1. Okay. Now, so what the things you have to remember, first is the distance between the phosphates of two individual strands is 20 angstrom. The distance between two individual strands is 11.1 angstrom. The angle between the first carbon and the nitrogen of the nitrogen base is 51 degrees. The molecular weight is 106 to 109 daltons. The protein histone occupies the major group at an angle of 30 degrees. Okay. Histone proteins are rich in histidine, okay, and arginine, which makes the nucleoprotein or nucleosome model. Okay, 3.4 angstrom is the distance between two steps. Total step of 10 total length of 10 steps is known as what helix length, which is 34 angstrom. Types of DNA. Types of DNA depending upon the direction of twisting. Okay, direction of twisting means whether they are twisting like this, mean clockwise or anti-clockwise. Or anti-clockwise. Right-handed DNA having clockwise twisting of the helix. Watson-Crick model of the DNA was the right-handed helix, and BDNA. BDNA is present in humans. Okay. Left-handed DNA, anti-clockwise twisting of the helix. Rich discovered Z-DNA model. Phosphate and sugar backbone are zigzag. Okay. In case of Watson-Crick model of type B DNA, which is present in us, is helical. The left-handed DNA is present in the in which the present in ZDNA, which was discovered by Rich, and the phosphate and sugar is present like a zigzag. Okay, understood? The right-handed DNA and the left-handed DNA. Okay. The types of DNA also include A, B, D, Z. All these types are given, and you will be, considering in the molecular basis of inheritance all these types. Ribonucleic acid or RNA. Configuration of RNA. RNA is fundamentally the same as DNA in the structure. However, there exist some differences, like deoxy sugar in DNA is replaced by the ribose sugar in RNA. I have given you the difference as well. Thymine in DNA is replaced by uracil in RNA. RNA is single-stranded with some loops, which are also made up of only one polynucleotide chain. Okay, RNA is single-stranded, made up of some loops, which are made up of only one nucleotide chain. Okay, some are the exceptions, like RNA found in reoviruses are double-stranded, two polynucleotide chains. RNA which are found in reoviruses are the exceptions, as they have two or double-stranded RNA. Okay, they have two polynucleotide chains. Types of RNA: genetic RNA or genomic RNA. When the DNA is not available in the cell, RNA works as the genetic material and even as gene for protein synthesis. Example: reovirus, tobacco mosaic virus, and QV bacteriophage. So our reovirus, tobacco mosaic virus, and QV bacteriophage, they have RNA as a genetic material. That is why this RNA is known as what? Genomic RNA or genetic RNA. This case happens when DNA is not available in the cell and RNA works as a genetic material. Understood? Non-genetic RNA or non-genomic RNA. What does you mean? And what does that mean? Non-genetic RNA or non-genomic RNA? I mean, this RNA will not exactly take part in the transcription process. It will not, help in the transferring the information from one generation to the next generation. So this non-genetic RNA or non-genomic RNA is not the hereditary material, but it will act as a tool. Indirectly, it will help in the expression of features like ribosomal RNA, transfer RNA, and messenger RNA. So first is the ribosomal RNA. Ribosomal RNA, also known as rRNA, 80% of the total RNA in the cell. The total amount of RNA which is present in the cell, out of which 80% is ribosomal RNA or rRNA. Kunz discovered rRNA. Kunz, for the first time, he discovered rRNA. Present in ribosomes and produced in the nucleus of cell. It is present in the ribosome and it is present in the nucleus of a cell. Mean these rRNA, they are making ribosomes, they are present in ribosomes, or they are produced in the nucleus of the cell. Okay. The most stable form of RNA. This is the most stable form of ribonucleic acid. Eukaryotic cells have 80S ribosome. S stands for Sidworth's coefficient or sedimentation coefficient, who has 60S and 40S subunits. The 80S ribosome in case of eukaryotes is further divided into smaller subunit of 40S and larger subunit of 60S. This type is made up of 4 different units of rRNA. Okay, so this type in 80S ribosome in case of eukaryotic cells, it is made up of 4 different units of ribosomal RNA. 4 different units of ribosomal RNA. Prokaryotic cells have 70S ribosome, who has 50S and 30S. 50S as a small unit and 50 and 60S have large unit. They are made up of 3 different types of units of RNA. They are made up of 3 different types of units of rRNA. Is it understood? Yeah. Function. They help in protein synthesis. rRNA provides the attachment site for transfer RNA and messenger RNA, which remain in the ribosome. Salt linkages are present, bonds. Okay, they are the bonds that attach transfer RNA to the larger subunit of ribosome, while the mRNA to the smaller subunit of ribosome. So ribosome, it is a site for the attachment of the transfer RNA as well as messenger RNA. Messenger RNA attached to the smaller site of subunit of ribosome, and transfer RNA attaches to the larger subunit of ribosome. Is that understood? Yeah. So this is the non-genetic RNA or non-genomic RNA, which is not involved in the transferring of generation, which will not act as a genetic material, but indirectly they will help in the protein formation, the translation process. Transfer RNA or tRNA, they are the part of 10 to 15% of the total RNA content, and total amount of RNA which is present inside the cell. Transfer RNA around makes 10 to 15% out of it. They are synthesized in the nucleus or on DNA. They are basically synthesized in the nucleus on DNA. The transfer RNA. They are also called soluble RNA or adapter RNA. They are also known as soluble RNA or adapter RNA. Hogland, Zemenke, and Stephenson discovered tRNA. Hogland, Zemenke, and Stephenson discovered tRNA. Hogland also gave the clover leaf-like model, clover clove-like model of tRNA. Single-stranded RNA, it is single-stranded RNA. However, the strand loops out to form a hairpin loop-like or clover-like structure. Okay, so it is a single-stranded RNA. However, the strand loops out from hairpin-like, okay, or a clover-like structure. Okay, like this. Like this. Okay. Function. During protein synthesis, it carries the amino acid to the ribosome. During protein synthesis, mean translation. Protein synthesis is translation. It carries the amino acid to the ribosome where it is made in polypeptide, when where the protein translation will happen. Messenger RNA or mRNA. Okay, mRNA, it is present or it forms only 1 to 5% of the total cell RNA. Huxley, Walkin, and Astrin discovered messenger RNA. As the name suggests, messenger RNA helps to transfer the information from DNA to messenger RNA. And this messenger RNA come outside the nucleus in the cytoplasm to attach with the ribosome and the tRNA for further processing. Okay. While it was named by Jacob and Monod. Okay, so messenger RNA was name given by Jacob and Monod, but discovered by Huxley, Walkin, and Astrin. mRNA is produced by genetic DNA in the nucleus. This process is known as what? Transcription. It is the least stable RNA. Most stable RNA is ribosomal RNA. Least stable RNA is messenger RNA. It also acts as a template RNA for protein synthesis. It also acts as a template RNA for protein synthesis. So this is all about the nucleic acids. Okay, let's see once again, what are the different slides content we have covered. Yeah, nucleic acids, DNA, its configuration, pyrimidines, purines, pentose sugar, the attachment of nitrogen base on the pentose sugar, the attachment of phosphate group of the pentose sugar. Okay, then nucleoside, nucleotide, the difference between nucleoside and nucleotide. Okay, all these are nucleoside and nucleotide differently arranged. Okay, you can see in case of the RNA, second position is having a OH, and in case of DNA, second position is not having the OH. Oxygen, sorry. Deoxyribonucleic acid. Deoxyribonucleic acid was discovered by Mischer. He discovered the DNA while the term was given by Sharis. The pentose sugar is deoxyribose sugar, and the nitrogen base are of 4 types: A, T, G, and C. Wilkins and Franklin, he studied the DNA. They studied the DNA with the X-ray crystallography technique, and Watson Crick also proposed the double helical model with the same technique. Watson, Crick, and Wilkins were awarded the Nobel Prize in 1962 for the same. Okay, DNA is made up of two polynucleotide antiparallel chains. The phosphate backbone of the phosphate group, they are present on the antiparallel side, and the nitrogen bases are complementary to each other. Okay. Then Shargat's rule, I told you, the base ratio, that is A plus T upon G plus C, remains constant, and A plus G upon T plus C, purines upon pyrimidines, it is equal to 1. It is equal to 1. A is equal to T, G is equal to C. This is the sugar phosphate backbone or the formation of polynucleotide. Okay, you can see the end which is free. The fifth end is free. The fifth end is free. This is 5' end. Okay, because the third end is involved in making the ester linkage. Okay, with the second nucleotide. So the OH of the third and the phosphate group of the fifth carbon, they combine to form the phosphodiester linkage. Facts: A plus T is greater than G plus C, then the DNA is AT type. If G plus C is greater than A plus T, then it is GC type. GC type means more TM, more melting temperature. Prokaryotes have more melting temperature as they are rich in GC type. Okay? Then denaturation of DNA, renaturation of DNA, hyperchromicity, hypochromicity, configuration of DNA molecule, length, distance, angle, everything is mentioned. Okay. Single stranded in case of the 5 into 174 bacteriophages, G4, S4, S13, M13, like that. Types of DNA: right-handed, left-handed, right-handed in BDNA, left-handed in case of ZDNA. Ribonucleic acid types: genomic, non-genomic. Okay. Then transfer RNA, messenger RNA, and rRNA. So, my dear students, we have completed the nucleic acids. In the next class, we will be starting with enzyme, which is a very huge topic of this. Uh, then after enzymes, we will discuss about some pointers about vitamins and minerals. Till then, bye bye. Take care. Stay safe. Stay healthy. And the most important is happy Deepavali. Okay. Bye bye.